but I will present to you some data today why we think that the blood-brain barrier plays indeed an important role in the progression of Alzheimer's disease, and I would like to introduce to you maybe a model how we can deliver drugs across the blood-brain barrier into the brain of AD patients. Well, we're not that far, but deliver drugs. As I stated before, Augusto Data was the first patient of Alzheimer, and when he was drawing these images, I mentioned that he was drawing these tangles in the neurons, and he was drawing these plaques. He was drawing a lot of microglia. Basically, 80% of all his drawings from her brain were microglia, but he was drawing blood vessels as well, and they looked a little bit different. And nowadays, when you look into Alzheimer's disease brains from a different point of view, from the point of view of the Braak stages. Braak is a neuropathologist who went through through thousands of brains of Alzheimer's disease patients, and he staged the pathology in these brains due to different markers. And one of the markers is a pathology within blood vessels, and I'll show you that in a minute. And I still believe that this pathology is driven through the A-beta peptides, which, at least to me, play an important role in Alzheimer's. Okay, so here again we have brain sections of a healthy young brain and of an old brain. And here are stainings of that you've seen in a different way before. The black staining in the brain of an AD patient, and this here is a blood vessel of an Alzheimer's disease patient. And most of the AD patients, if not all, show something like this. It's called CAA, cerebral amyloid angiopathy, meaning you have deposited A-beta around the blood vessel in the brain. And you can hardly see that here, but it should show you, here you have a blood vessel, and right on the blood vessel you have deposited A-beta plaques, basically. So apparently A-beta is not only deposited centrally in the brain, but at the blood vessels themselves. So the blood vessels mean the blood-brain barrier in the brain. What is the blood-brain barrier? The blood-brain barrier was first introduced, or the term was first introduced by Paul Ehrlich in 1885, and he did a really excellent, easy experiment to show, or he wasn't looking for blood-brain barrier, so what he was doing basically is he injected a blue dye into an animal. And then he sacrificed the animal and just checked where the dye goes. And everything inside the animal was blue, except the brain. The brain was not stained. Then he said, oh, that's somehow weird, because there are blood vessels there too, so the dye should go there too. Then he injected the dye into the brain of an animal, sacrificed the animal, and now only the brain was blue, and the rest of the animal was not stained at all. So by then he realized, okay, there must be something specific about the blood vessels in the brain, and he called that then the blood-brain barrier. And it's really a very tight barrier. So when you look at the brain, or when I introduced to you the brain before, I always chose this picture. But to me, when I talk about the blood-brain barrier, it looks like this, because it's really the whole vasculature in the brain that is important for all different kinds of supplying of nutrition, for example. So the blood-brain barrier, or the vessels in the brain, are built, of course, by endothelial cells, like all the other vessels in our body as well. But these endothelial cells are not completely, but they're different to all the other vasculature that you have. Because here we have cells that attach to each other, that is normal. You have always tight junctions in endothelial cells everywhere in your body, but in the blood-brain barrier they are much tighter. So they are so tight that you really have almost no diffusion from here, from the inside of the lumen of the vessel to the outside into the brain. So this is one hallmark of a blood-brain barrier, that the vessels are extremely tight. And you have not only endothelial cells, on top of it you have pericytes that are next to the endothelial cells, to the basal lamina, and around there are astrocyte end feet that cover this structure, this neurovascular unit, to say so. So that is the structure of this vessel. And then you have here small receptors or transporters, which when we magnify them, you can see that they all have different functions. So for example, we have here PGP transporters. These are basically transporters that take stuff and traffic it back out of the cell into the lumen or back into the brain. So they don't transport a lot across, but they're basically recycling stuff back into one direction or the other. You have amino acid transporters, because amino acids cannot just diffuse across the blood-brain barrier. It's far too tight. I mean, you have a lipid bilayer on both sides, and you have no space between the tight junctions that anything can diffuse to. You have glucose transporters to transport glucose into the brain. Nutrition, everything. Everything has to be transported. And you have, shown here, bigger receptors, different receptors that can take up all kinds of ligands and transport them specifically out of the brain or into the brain. So only lipid-soluble molecules can basically diffuse from the blood into the brain, and only if they're really small. The bigger they are, you need already, again, specific transporters. Just a water-soluble dye, as Paul Ehrlich has shown, just is not going across this tight monomer. And this is just due to the fact that you have tight junctions, which are very, very tight. Okay, what is going on in Alzheimer's disease? And I took this out of a publication by Berislav Slokovic, and what he stated is the following. Up here you have an image of the endothelial cell here in red, with the basal lamina around there, astrocyte end feet, some pericytes here, neurons in the brain, some microglia, and here some A-beta oligomers. And in the early phase, it all looks pretty normal in the brain, and the later you start to get early symptoms in Alzheimer's disease, you have even more A-beta oligomers, and you see already some structural changes in neurons maybe, and you see some structural changes already at the blood vessel, very early on. Then you have a late symptomatic stage, and there now you have fibrils of peptides, and the blood vessel is completely shrunken, and in the end stage, there's a complete breakdown of the blood-brain barrier, so a complete opening of the blood-brain barrier during Alzheimer's, or an end stage of Alzheimer's disease. The question really is, when do we have that opening? Is that opening, this is certainly correct, but do we have here already some opening, or do we here have already some opening of the blood-brain barrier? We don't know, we have no clue. Okay, so what is A-beta now doing? He put that already in that image. A-beta, here shown by another publication, these things here are A-beta plugs at blood vessels. So it seems that they really attach to the blood vessels, and then if they attach there, they might break up these vessels. It's an idea, it's not proven, okay, at least to my knowledge, but maybe it could happen in this way. So if A-beta is then this problematic, how is the brain getting rid of A-beta in general? There are different ways how the brain can do that. So the first way that has been published, and it works pretty well, if you do that, these experiments, you can get rid of A-beta 1 to 40 or A-beta 1 to 42 when you put it together with enzymes like neprilysin or the insulin-degrading enzyme. These enzymes are capable of degrading A-beta peptides, and they're expressed in the brain. So part of the A-beta is all the time degraded by these enzymes. Another way to get rid of A-beta in the brain is through uptake of glial cells or neurons, for example, and this usually goes through the LRP1 receptor. that has been shown by Journal in a very nice paper where you could show that A-beta directly binds to LRP and is then degraded. He has shown that in neurons. Or A-beta, you can get rid of A-beta if you just form plaques, right? So one way could be, well, we know already that the plaque pathology does not go along with, let's say, the amount of plaques doesn't tell you whether you have a severe Alzheimer in front of you or only a mild Alzheimer in front of you. So plaques do not correlate with the disease very well. So therefore we believe that the plaques are kind of way of storage of the beta peptides if you can't get rid of them. So that would be another way to get rid of the beta peptide. Or, another way would be through clearance across the blood-brain barrier, so basically across the endothelial cells into the bloodstream and then into the liver, and there it's degraded. As you know, A-beta is generated theoretically all over the body, since APP is expressed everywhere, all the enzymes are everywhere, but we do not have plaques in the muscle or in the kidney or wherever, because A-beta goes into the blood, goes to the liver, and is degraded. And this might happen here as well. A-beta, again, most likely binds to LRP and is then transported into the blood, and we should be able to get rid of it. So this part was introduced a couple of years ago by Berislav Slokovic and his group, and he has shown pretty nicely that LRP is a very good candidate to do that. But due to the fact that he had no genetic model in hand to prove that LRP really is the key receptor, there were still some questions out there whether it's LRP or other receptors that are doing the job. So we started out by then and were focusing more and more on LRP and trying to use genetic models to prove that LRP plays a key role in the clearance of the beta peptide across the blood-brain barrier. Okay, LRP, as I said before, is a member of the LDL receptor family, and all of these could theoretically play a role in clearance of the beta peptide. Some of them have been shown to bind the beta, but this one for us was the most likely candidate. Okay, so yesterday I told you already in very early experiments that I did in 2000, we were able to see that if you just add a ligand, alpha-2 microglobulin, to regular tissue culture medium where A-beta is in there, the alpha-2 microglobulin binds to A-beta and then is internalized through LRP1, since we could block that with RAP here, or that LRP knockout cells had no such an effect. Okay, so that was clear. A-beta can somehow be internalized or transported through LRP. To transfer that into an AD model, we checked those days, and I mentioned that before, that LRP is indeed downregulated, as you can see here, in Alzheimer's disease patients. And this here is a quantification Dave Kang did those days with 39 patients, age-matched for normal healthy controls, age-matched, and Alzheimer's disease patients. So you have a dramatic reduction in LRP expression. But this is whole brain of an Alzheimer's disease patient. Then here was a publication by a different group. They made an LRP staining in vessels and in mice. And the older the mice got, the less LRP expression you've gotten as well. So another good indication that over time you lose LRP expression. This, but here only shown in mice. Okay, so then we're back to our question. Is LRP expression good or bad? If we have high LRP expression, I told you yesterday, then we have an increased A-beta production. Or when we have a high LRP expression, do we have increased A-beta clearance? And we wanted to answer that and started very simple with some tissue culture experiments. We wanted to use a in vitro blood-brain barrier model. And to generate an in vitro blood-brain barrier model, it's actually quite easy. What you use is you have these chambers here where you usually have your culture dish with your regular media, and you have an insert in this dish with a filter in here, and on this filter you grow your endothelial cells. But we tried a lot of endothelial cells which are just regular cell lines. If you can do that, it's not a problem at all, but these are not real endothelial cells. They lose a lot of their characteristics when they are cell lines. So what we always did now was we generated primary mouse brain endothelial cells to get this monolayer in a correct way. And we chose primary mouse endothelial cells over porcine or bovine or whatever, because there we could use all kinds of different genetic models to get the endothelial cells. So if you have this, then you have to make sure, you have to make 100% sure that you have here a monolayer, a really tight monolayer. If you have one opening here in between, everything can diffuse and you cannot measure any kind of transport at all. So therefore, we went, after painful learning that, to a company who sold us this machine here, and that is actually a quite nice thing. What it does is the following. You have the possibility to measure the impedance and the ohms resistance over the whole area. Meaning, the ohms resistance tells you how tight some barrier really is. And you can measure online in the tissue culture incubator the ohms resistance. So at some point in time, that goes up. So here you have a tight monolayer that tells you the impedance as well. And then at some point in time, you have really a certain resistance. Then you know you have a tight monolayer. Then you can start to really measure transport across a tight monolayer. A lot of people use just chopstick electrodes. So what they do is they do one chopstick here, one chopstick there, and then they measure the ohms resistance and get some kind of value. But the value they get is from exactly this point here, right? If you move your chopstick to here, you get a completely different value. So when you look into the literature, some people come up with fantastic so-called tier resistance values, and they just move around their chopsticks as long as they have a nice data set, and then fine. But when you do this, you cannot cheat, right? I mean, it's like it is. But this is a real problem, I can tell you that, because we just today, another paper from us was accepted with this method here, and we had to fight one reviewer who always told us, all your results are completely bogus because your resistance is far too low. And then we had to go through half a year using all different kinds of methods to show the editor and the reviewer that we can of course cheat and get very high resistances when we use chopstick electrodes or other methods, but with our method it always stays the same. The values are lower, okay, but these are the real values. And so it was really painful. But one reviewer, I bet you, measures all the time with chopstick electrodes. That was, yeah, well, that was a moment. But anyway, we got through. Okay, so this is a way to build up an in vitro model, an easy in vitro model. You can do that much more sophisticated for blood-brain barrier. So what did we want to investigate? We wanted to see whether A-beta peptides are really transported across the blood-brain barrier. So therefore, as I said, we have here our endothelial cells, our monolayer, and we just assume that the part up here is our blood compartment, where you have your blood lumen, and the lower part here would be the brain. So we want to measure the transport of A-beta from the brain into the blood. Therefore, we just added to this compartment radiolabeled A-beta peptides, and we added them at a concentration of 0.1 nanomolar. That is very little A-beta, and that is the physiological concentration of the A-beta peptide. When you look into the literature, most people use A-beta in micromolar ranges. You will never find that in the brain. So this is due to the fact that we could use radiolabeled A-beta, we could go that low. Additionally, we added always C14 inulin. So we added this because inulin has no known receptor in vertebrates. So when we add inulin in the beta, what we do is we measure the input here, and we measure how much is getting to the other side. If inulin reaches the other side, it's diffused to the other side. So there is no receptor for it. And then we can basically subtract the amount of inulin that has reached the other side from the amount of A beta that has reached the other side. And the difference then is receptor-mediated transcytosis. Okay? And we use, of course, from time to time RAP to block the transport from here to there. So what happens is, when we just use, when we measure the transport of A beta in comparison to inulin, we see a very fast transcytosis to the other side, which to our surprise then plateaued. So I would have assumed in the very beginning we add some A beta here, and at some point in time over time we get more and more and more over here until we have a concentration equilibrium between this one and that one. We most likely don't reach that because we have the plateau quite early. And we can block that with RAP when we add RAP here at the same time, so A beta is not as efficiently transported. Okay, so this, first of all, tells us not all A beta is transported by LRP or by lipoprotein receptors, because RAP can't block all of it, but a lot is transported by lipoprotein receptors. And it tells us there might be some other effect going on, not only transport from here to there, but maybe transport from here back into the brain. So to analyze that, we used again our knock-in mouse model with the knock-in in the second NPXY domain of LRP, and that meant that these LRP molecules are not endocytosed, and if they are not endocytosed, they are not transcytosed. So transcytosis is completely reduced in these animals. So therefore, we to show that on a genetic basis that LRP is really involved in that transport and not all lipoprotein receptors, we repeated the experiment, and you see that we have an approximately 50% reduction in transport of A beta peptides from the brain compartment to the blood compartment when we just use the NPXY to knock in endothelial cells. So 50% of the A beta is transported somewhere different by other receptors, which is okay, but 50% is just by LRP1, nothing else. So then we wanted to know, okay, is there retransport from LRP to this site? Because there is a publication, or there are many publications out there, showing that another receptor, you can hardly see that probably, which is called RAGE, it's a different receptor, has nothing to do with lipoprotein receptors, is capable of transporting A beta from the blood side to the brain side. Well, we had some troubles to detecting RAGE, so therefore I thought, does it really have to be RAGE that is transporting A beta back? Or, if you think about it, you have a molecule that is supposed to transcytose A beta from here to there. How is it doing it? It captures maybe A beta and then transports it in vesicles through the cell, and it has to fuse to the other side of the cell to release the A beta peptide. Most likely LRP is still there. So if LRP is still on the other side, shouldn't it be capable of capturing A beta from that side and bringing it back into the brain? And we, of course, tested that. It was easy for us to test. Again, exactly the same setting, A beta, inulin, RAP, GST, and unfortunately, LRP is capable of bringing A beta back into the brain, at least in an in vitro system. So, whether that's good, I doubt it. So, the question is, which we have to answer now soon, whether that happens in vivo too. We are at the moment in the in vivo stage, and I'll show you results about that now, but we haven't tested the transport from the blood into the brain yet. But what we have done is we have generated a new conditional knockout mouse model for LRP, and therefore we used the original LRP flux mice, which were generated by Joachim Herz, and when you just induce the knockout, these animals die, as I said, E10 if you induce a knockout. And we crossed them into a mouse that has a specific promoter which is specifically localized in the mouse brain endothelial cells. So no other cell is affected or should be affected when you use this promoter. When we crossed these mice, we got our novel LRP conditional knockout mice, which we can now induce whenever we like with tamoxifen to have a knockout in the blood of LRP1 in the blood-brain barrier. To show you on a genetic level that that really worked, we have a PCR strategy to show that we have either here complete recombination when we have the induction of Cray, or no recombination when we have no Cray induction. Okay, that's just PCR for screening. Here now some data that shows you that these cells really do not express LRP. So when you generate primary mouse brain endothelial cells, it's really a messy action. You get these kinds of bundles here. This is what you start out with. And then you would probably immediately say, oh, well, wait a second, that's not only endothelial cells, there is some other stuff in there. So how can you be sure that you really have only endothelial cells in there? That's really easy. Endothelial cells have one nice thing. As I told you before, they have an ABC, they have the PGP transporters. PGP transporters are fantastic because what you now do when you have these cells in culture, you add to your culture medium puromycin. Puromycin is an antibiotic which kills all your cells except endothelial cells. Because endothelial cells, through that PGP transporter, throw out puromycin all the time. So they survive, and all the other cells you have in there die. Very easy way to get a 100 pure endothelial culture, like you see here after 5 days. And when we lyse these cells then, and this here on this side is a control setting where we have either LRP expressing cells or knockout cells, or our primary mouse brain endothelial cells. And when we induce the Cray, so we have a knockout situation, we have no LRP expression. So that is on a biochemical level. Then we did, of course, stainings for LRP in these cells, and you probably can see we used a marker for endothelial cells, and here you see one vessel basically stained with this marker. Similar staining with LRP1, and in emerge you see nicely in yellow that you have a really cool colocalization. If it would be darker, you could see that you have some background stain here in red everywhere. And that is good, because that means, this is a brain section. That means that in your astrocytes, in neurons, you still have LRP expression, right? This is, my mistake. This is the not induced knockout. So there is no knockout, of course. Here is the knockout now. You still have the vessels, but you have hardly any LRP staining in the vessels. But you have background staining everywhere here, which is regular LRP staining in astrocytes and neurons. And you have no co-localization of LRP with the endothelial marker. So we really have a knockout in our endothelial cells of the brain of LRP. This is just a different way of cutting in the larger vessel here now, and you nicely can see that you have here, when you have no induction, you have a nice co-localization, but in the knockout there's no LRP expression in the vessel. Okay, so we have that mouse model now. The first thing then we have to prove is, are these mice, do they really still have a tight vasculature? Or does the knockout of LRP affect, in general, the structure of the brain vasculature? And to prove that, we just injected antibodies, if you wish, into the periphery of these mice, and then you check in the brain how much is in the brain, in general, how much is getting in. And whether you induce or not induce the pre-promoter, or whether you have LRP there or not, doesn't matter. Same amount of antibodies are there. So no difference. We did that many times, and there's no real big difference. We did that with a fluorescent marker as well. Again, no big difference. So in general, these endothelial cells, or these neurovascular units in the brain of these mice seem to be normal. Then we took primary endothelial cells from these animals, plated them again in our tissue culture system, and you can see right away that the transport of A beta from here to there is reduced in our knockout animals to almost 50%, as you have seen before for delta and PXY2 knockouts, right? So again, very good proof that 50% of A beta in vitro is transported by LRP. And this is after 45 minutes, this is after 24 hours, so that is pretty much, works pretty well. Then we said, okay, now we really have to go into the animal. And therefore, Stefan Stork, a PhD student of mine, bought the stereotactic machine here and started to stereotactically inject mice with A beta peptides, with very little radio-labeled A beta peptides. So what he was trying to see is, he was interested in whether these A beta peptides in a living mouse would reach the bloodstream in either knockout LRP or not-knockout LRP. And this is what he saw after stereotactic injections. So you see here in black the LRP knockouts, if I see that correct. Yeah. And here you have the flux mice. And this is the remaining of the A beta in the brain. Okay? So in the knockout, when he injects A beta, you have a lot of A beta remaining. And when you have LRP, 50% of the A beta is cleared. And that is just, this is at the same time, in the same syringe, he was injecting inulin. Right? And inulin, there is no difference between the two phenotypes, either knockout or LRP expressing cells. So this, again, is a specific effect now in a living animal of transport of A beta 42, actually, out of the brain. So the toxic form of A beta. So after he did a lot of calculations, he came up with this number and claims that from the total brain clearance, LRP in endothelial cells clears 26% of the A beta, only of endothelial cells. LRP is in microglia and neurons, clears as well. But across the blood-brain barrier of endothelial cells, of the total number is 26%, and clearance of the blood-brain barrier in total is almost 50%. Okay? So then we said, if this is correct, we have to see a phenotype in Alzheimer's disease mice. Alzheimer's disease mice means there are different mouse models around there which should reflect Alzheimer's disease at some stages. It's not so easy to do that, and what we call Alzheimer's disease mouse is really not a mouse that has Alzheimer's disease, but it has some features. So we always look for plaque development. Ideally, we want to see neurodegeneration in the hippocampus, and on top of it would be really good to have a nice behavioral readout that these animals cannot remember as well. So learning and memory should be impaired in these mice. And this is such a mouse model, and it has a nice feature. It has 5 mutations in APP in the presenilin gene, produces a lot of A beta, has very fast plaques, has a severe phenotype, and we use these mice and cross them in our LRP inducible knockout model. And first checked what happens to A beta in these mice. So I have to say, these mice, the promoter of these mice is exclusively expressed in the brain, so not in the periphery. Okay? So every A beta that is generated is generated in the brain. So when we now check for A beta in the plasma of these animals, you see that in the knockout animals, you have hardly any A-beta coming from the brain into the blood. But the mice that do express LRP transport nicely A-beta that is generated in the brain into the bloodstream. Then we ask, okay, how about the A-beta that is remaining in the brain? The soluble A-beta that is remaining in the brain. Here again, knockouts, you have a lot of A-beta remaining in the brain compared to LRP, just the LRP expressing mice, more A-beta is getting out of the soluble form. And then we tested the insoluble A-beta, which is supposed to be A-beta that is deposited in plaques. And here we have in young mice pretty much the same, that you have, when you have no LRP, A-beta can't leave the brain, you have more deposited in the brain compared to mice that have LRP. Okay, so when we did quantifications then on an ELISA-based assay, we saw pretty much the same. In young mice we have less clearance of A-beta 40 and of A-beta 42 when you knock out LRP in the bloodstream of these of the blood-brain barrier. So then we tested plaque load themselves, and you see right away in young mice these are not induced, and these are induced. You have much more plaques, or many more plaques, and they are localized closer to the vessels. That was interesting to us. So it seems to be that LRP has some effect on a vessel phenotype in accordance to A-beta deposition. When we looked at older mice, it looks still good because we see still a huge difference in soluble A-beta peptides. So this is 40 and this is 42, so there's a dramatic drop or dramatic difference in the clearance of the A-beta peptide in the knockouts. But when we look at insoluble A-beta, the difference is not as big anymore. So that means probably these mice deposit still A-beta. We still have similar amount of plaques suddenly, but the soluble form is very well cleared. So that should mean that if the soluble form is the toxic form, these mice, when they're getting older, should behave better than but he's not induced. Okay, oh, sorry, this is the plaque load of the old mice. There's no big difference in the plaque load, unfortunately. Or fortunately, I don't know. But that's what it is. We have gliosis in these knockouts, so we have a lot of A-beta remaining, so we get huge gliosis in the brain, which is kind of to be expected. And we tested that, of course, now memory in these animals. And to test memory, our collaborative partner, Thomas Bayer, who did all these testing, always uses these tests here. What he does is the following. He has these trials where he has some cues here around the Morris water maze, and in this area there is a platform. So he puts the mouse somewhere here, and the mouse has here some cues and can remember, aha, the platform has to be in this quadrant here, so I'll swim there and sit down on the platform and everything's fine. Then what he does is, then he removes the platform, and then he measures where the mouse is swimming around. Is the mouse directly swimming to this platform, or is it swimming in circles like here? Can't remember where the platform is, or is swimming more directly to that quadrant here. So when you look at this here, the animals where LRP is not induced, so you still have LRP expression, the mice swim almost directly to the area where the platform was, and not so much to the others. But when we knock out LRP, they have no clue where to swim. They just swim around here. Okay? They cannot remember where to go. And their swim speed in general was pretty similar. We have that paper in revision, and the reviewers asked us to do more behavioral analysis because our analysis was really only very poor and we had to do more. And I have not included that yet because it's not finished. But we have done another 5 different assays, and in all assays so far, these mice scored really, really poor. And this is astonishing because this mouse model, the 5XFAD Alzheimer's disease mouse model, is pretty much the worst mouse model you can get for the animals, because they get such a severe phenotype, and we were even capable of making them die. So that is really hard. I mean, they develop it so early and have such a strong phenotype that we want to do these experiments again in a not so severe mouse model, but that will be so expensive that I think about it. If we get that published this way, probably won't do it. But we'll see. Okay, so so this is the story about what I believe is important in the blood-brain barrier, that certain transporters are capable of transporting A-beta out of the brain and should be able to clear A-beta from the brain. LRP clearly, in vitro and in vivo, has the capacity to clear maybe 30% of the total brain A-beta across the blood-brain barrier, and that is already a lot. Okay, let's stop here, and in the second part we will talk about some possible therapeutic strategies that we can use with our knowledge of the blood-brain barrier. Okay, thanks. We analyzed so far the transport from the brain, out of the brain into the bloodstream, and these mice seem to be fine. Now what we at the moment are doing is we are trying to find out whether A-beta can be transported into the brain, but we haven't really done any experiments on that yet, but we will. And then that might, well, hopefully answer the question whether A-beta is a kind of a seed for plaque pathology, and maybe that seed can even come from the periphery, as many people have shown. Matthias Jucker's lab has shown that, for example. And maybe this mouse model will help us to analyze whether LRP is a transporter for such a seed across the blood-brain barrier from the blood side to the brain side. But so far we have no data. We haven't even started the experiment, but we will do that. So now I want to continue with some other ideas that we have more in regard of therapy. It's not really therapy, maybe it's therapy of mice and not of humans. But what we want to do is, for example, if this is correct, the idea that A-beta can be transported from the blood side into the brain, then we should be easily able to block that transport. For example, if you think about it, if we just generate an antibody against LRP and inject that antibody into the bloodstream, it should bind to LRP in a way that A-beta can't bind anymore. Therefore, you would transport less A-beta into the brain, but still would maybe transport A-beta out of the brain. That's just the basic idea. You could say, well, stupid idea, because if you have an antibody there, that antibody is transported to the brain and then it recycles all the time. Maybe, I don't know. We have to see that. But we want to follow that up and generate these antibodies. So what we did is, since LRP is such a huge molecule, we just took small pieces of LRP and made monoclonal antibodies against these pieces. And many medical students of mine went through the painful work of characterizing hundreds of different monoclonal antibodies and pinpointed the binding site of these antibodies to very small fragments, so that we are now down to 10 amino acids where we know exactly where the antibody binds in LRP. Different antibodies we've generated, and these antibodies we are at the moment trying to figure out whether these antibodies block the binding of the A-beta peptide to LRP and thereby cannot be internalized anymore. And this is one example here where we tried to block the uptake of A-beta, radiolabeled A-beta, in the presence of these antibodies. So this is here just a control. this is, well, these are two controls. When we add the antibody to our cells, to living cells, together with A-beta, we have here, let's say, 30% reduction in uptake of A-beta. And then when we titrate the epitope that we have used, which we have mapped where the antibody is binding to, and we titrate that in bigger amounts into the tissue culture medium, we can compete for the binding of the antibody to LRP, and therefore get again complete restoration in the uptake of the peptide, of A-beta. So this is a good indication that the antibody theoretically might work, and we will use these antibodies when we have established that A-beta can indeed cross from the periphery into the brain to inject these antibodies into the animals and see whether that then can block transport across the blood-brain barrier. That would be cool. But assuming this is correct, we can all speculate about it. So let's assume, just for the sake of argument, whether it's correct, I don't know. Let's assume 50% of the A-beta in your brain comes from your periphery. Okay? And I'll tell you, you get my antibody, I inject it in your bloodstream, so 50% you have 50% less transport from the periphery into your brain. Therefore, you shouldn't get Alzheimer's. Would you do that?
What do you think would happen with all the endothelial cells when LRP is blocked? Really good.
Endothelial cells.
So, at risk of...
Yeah, might be a problem, but I don't know. Really, endothelial cells is your biggest worry? You know?
The risk factor for atherosclerosis.
So, well, I would even say, hey, that's even better because I blocked the uptake of LDL. So I should not even have a problem with that anymore.
Yeah, how about monocytes and macrophages?
Monocytes and macrophages. Yeah, might be a problem. Well, I don't know. I have something completely different in mind.
Well, can you use it for whole life?
I don't know. But I'm just talking about a single injection. Would I take a single injection of an antibody against LRP? No way! Highest expression level of LRP is the liver. If you kill LRP in your liver, what you basically would do, right? You have no uptake of A-beta in the liver anymore, so it circles around in the blood anyway, and you have no uptake of HDL in your liver anymore. Forget about it. I mean, that's probably the fastest way to kill you. I mean, maybe not. I don't know. But I still think, I mean, you have to think of all the consequences when you do such an experiment. But I mean, we'll do the experiment anyway. Maybe it's not correct what I'm saying, but maybe it works. I don't know. But we will see. But at the moment, I'm not so sure. But still, I think it's a cool idea. We'll see. Okay, so when we then did some kinetics on the overall transport from all the data we had from tissue culture and from the animals, we calculated that LRP is capable of transporting, theoretically, around 65 nanomole A-beta per hour. Because we come up to that number since the area of your endothelial cells in the brain is at least 25 square meters. Some people say it's even more. And when you calculate then the amount of LRP on 25 square meters in regard to the transport that we see, then you come up with such a number. If that is then correct, if this is correct, then you could clear your whole A-beta out of a brain in one day. If that is correct. Okay? So we will see whether this holds true. So now back to therapy in a different way, but again in regard to the blood-brain barrier. the very first day, I told you about the story of NSAIDs, right? That NSAIDs have a maybe protective effect because they modulate gamma-secretase activity. If you use different NSAIDs, like here ibuprofen, you reduce the amount of beta-42. And this was not proven, but it was shown in the Rotterdam study that people who take 3,200 milligrams of NSAIDs each day over many years, they have an 80% reduction in the risk of developing Alzheimer's disease. But again, the people who weren't here the first lecture, do not do that. Okay? Do not eat ibuprofen in those doses. It's really dangerous. So, fluorizan was then developed, a drug that looks like an NSAID, but does not touch the COX enzymes. But unfortunately, fluorizan, when you use it, does not cross the blood-brain barrier. So therefore, it failed miserably in phase 3 trials. So we thought, okay, we don't want to give up on this idea that we might have a drug that works. We just have to convince pharmaceutical companies to still look into these treatment options. So we thought, if this is not crossing the blood-brain barrier, maybe we have to deliver it in a different way. And for that, we tried to package flubiprofen or ibuprofen into nanoparticles. And what we used were polylactic acid nanoparticles, so completely biodegradable nanoparticles. So when you usually use flubiprofen and you add it directly to cells, this is what happens. You can use up to 300 micromolar of flubiprofen and your cells live happily ever after. But as soon as you go over that concentration, your cells die. So what we then did is, okay, we packaged our flubiprofen into nanoparticles and just added it to cells, and even to very high concentrations, our cells just lived at no problems with that concentration whatsoever. So that was good news, but this doesn't mean that it has any effect, it just means nanoparticles don't kill cells. Okay. Then we said, okay, what happens to our nanoparticles when we use them on our cells? And here we just measured uptake of nanoparticles that were loaded with flubiprofen. And we measured that here at 4 degrees. So at 4 degrees, your cells are pretty much silent. They have no basic activity, and you don't see endocytosis. So our nanoparticles can't be seen here. You would see them here as green dots, because you see them when you heat the cells up at 37 degrees, and these nanoparticles are taken up, and they were, of course, washed. The surface was washed away with an acidic wash, so you had nothing stuck to the cell surface, and this was all inside the cells that we have used. So we see uptake of nanoparticles into cells. Then we ask ourselves, okay, do these nanoparticles have an effect on the production of A-beta? What we use, therefore, is a cell line that stably expresses APP. And this cell line, of course, has the gamma-secretase complex and usually produces a lot of A-beta. And when we just add our nanoparticles into this culture medium with the flubiprofen, this should have the same effect as flubiprofen alone. So we have shown before, when you add just flubiprofen, that was that article I've shown you, you have a reduction in beta-42. And the hope for us was, okay, as flubiprofen is still active when it's in the nanoparticle. So the nanoparticle should then somehow act on gamma-secretase itself and reduce the amount of beta-42. We know these days already how these NSAIDs work. They really bind to the presenilin and therefore modulate the activity of the enzyme, and the enzyme cleaves at a different position and doesn't cleave at the position to generate the beta-42. So if this is correct, this should work. We first just added flubiprofen to our cells, and as before, we see a nice reduction. This is not so nice, a huge error bars here, but anyway, we see a nice reduction in A-beta-42. When we add flubiprofen with our nanoparticles, we see exactly the same. Not as such a strong reduction with low concentrations, but with higher concentrations. We see a nice reduction in Aβ42. So flubiprofen can be either released from the nanoparticle, or the nanoparticle itself with flubiprofen binds to gamma-secretase. Doesn't matter which way, we see a reduction in Aβ42. The question then for us was, would this work in a blood-brain barrier model? So again, we chose this time our endothelial cells on our filter system, and we plated at the same time in the lower compartment cells that express APP. So at this time, here in this compartment, there should be a lot of Aβ, right? Because it's always produced by these cells. And we just add the nanoparticles with the flubiprofen to the upper compartment here. And then measure whether the flubiprofen can cross the blood-brain barrier, and we see a reduction in Aβ42. Okay, flubiprofen alone in this setting, no effect, because flubiprofen can't cross. And that has been shown in the human trial already. But when we use the nanoparticles, we see a 25% reduction in Aβ42 in this setting. So theoretically, we could try that. So theoretically, this should be able to cross the blood-brain barrier. And this would be really nice if this is correct, because when you do the math on it, you need only 0.7% of nanoparticles to reach the brain to get a sufficient reduction of Aβ42 to have an effect, a possible effect on Alzheimer's disease. So why am I saying that? If you think about Alzheimer's disease has an exponential curve when you're from the age of 65. So from 65 on, your risk to develop Alzheimer's disease gets exponentially up, right? And this is most likely due to the effect of the amount of Aβ peptides that is there. So if you could be able to reduce the amount of Aβ peptides by 5, 6, 7%, not more, the age of onset would shift 5 years to an older age. And by that, you would reduce the amount of Alzheimer's disease patients by 30–40%. And that would be amazing if that's possible, right? Because then you would develop Alzheimer's disease. It would start exponentially with 70, 72, 75, I don't know. And then full-blown Alzheimer's would be reached when you're 90, maybe. And then you're dead already when you cross the street here. I mean, they almost killed me today. So that, if that is correct, that would be a really nice way to treat the disease in a prophylactic way, right? Because this drug you would have to take maybe 5–10 years before you get Alzheimer's disease. And then we are back to our problem. When are you getting Alzheimer's disease? I don't know, right? So, if we have a marker that would tell us you get it in 10 years, you could get such a treatment, not now, but maybe soon. And then the onset would be shifted 5 years. Okay, in your case, well, you're too young. But for older people, okay, that would most likely help them. So that would be a cool way to tackle this kind of disease. So this is what we are working on in our lab, and we will do these experiments now in the mouse model to see whether it works there too. And we have started that in a European-wide collaborative effort, and we were able to see nanoparticles in the living animal crossing the blood-brain barrier, no problem whatsoever, but we saw something else. So what we did is we loaded these nanoparticles with magnetic particles and could even see in an MRI exactly where they're going. I can come back again with my question. So everything I tell you here it sounds great, right? I mean, we solve Alzheimer's disease, great. Would you like that I inject nanoparticles in you? It depends, okay, on what?
They can be stored somewhere else.
Where?
The risk of side effects.
But where? Yeah, side effects, but they're lactic acid. I mean, it's great. What do you think? Where are the nanoparticles going? Liver. Everything goes to the liver. Where you have blood, it goes into the liver. Liver, spleen is full of it. I don't know whether I would like an NSAID specifically transported into the liver, although it's not working on COX enzymes. But side effects are completely, we have no clue what it does, right? So just adding nanoparticles to an animal, or you can do that, but I would never do that to humans, right? What we have to do is, we have to think about a cool way to tell the nanoparticle to just tackle the brain. So one way would be to get more into the brain, to fuse an LRP antibody to it, so that more binds to endothelial cells and gets transported. But then again, it gets even worse in the liver, right? Because there's even more LRP. So we have to find a molecule that is higher expressed in brain endothelial cells than in other tissue, and find a way to target that and then transport it into the blood, into the brain. One can think of maybe transferrin receptor or other receptors, but they are always expressed somewhere else as well. But that could be a way to more specifically target nanoparticles to the brain. So we are working on, of course, on the transport of A-beta peptides out of the brain. That works pretty well so far. And of course, we are working on the transport of drugs into the brain. And, well, that works in tissue culture. But I don't know whether that works in humans already. Okay, to finish this up, this work was, the nanoparticle work was done by Sabrina Meister in my lab, and she's just finished and left the lab. And Steffen Stork, he's still in the lab. He's doing the knockout animals and the transport of A-beta out of the blood-brain barrier. And this project was started by a former lab member, Thorsten Pflanzner. He's at AbbVie Pharmaceuticals now. And I really have to thank you very, very much. It was really kind of you, first of all, to invite me, that I could give this lecture. And I enjoyed it so much, all the discussions I had with you. That was really fantastic. And that you all came back, and some even new faces here. Thanks a lot. And if you have questions, now is the time. Thank you.
Okay. Are all the parts of the blood-brain barrier have the same resistance?
You mean everywhere in the brain?
Yes.
Whether it's a small capillary or a bigger one? Or you mean cortex or?
Yes, different regions.
Good question. I don't know. I don't know how to measure that. I mean, how would you measure that in vivo?
Maybe you should categorize.
Yeah, but I mean, No, I don't know. I mean, you could do that in vitro, right? I mean, you could isolate endothelial cells from specific areas, but that's in vitro, right? In vivo? In vivo. No idea.
I think there was a work with some fluorescent markers, some like luciferase that you can scan. And then they have seen in different regions that it's transported somehow faster
Yeah, but
in some
they're
regions.
they're theoretically not transported at all.
Or was something which could be transported. And then they scanned in vivo.
Yeah, but if it's transported,
Yeah.
if a fluorescent marker is transported, it's not diffused, right? But he's asking for tightness, and that is diffusion. But if a dye diffuses, then you have a problem.
So this
Dye
problem-
diffuses usually doesn't diffuse even in other organisms.
was something that was transported, I don't remember if it was a dye or something, to the method which was faster transported to one region so in the others.
Yeah, but was it in a disease case or was it in a
What's
normal
this?
condition?
I believe it was a normal condition. But it's about the conference, I don't remember.
Okay.
Check it out.
I don't know. But you had a question.
Yeah, I had a question about, in the very first lecture you proposed the treatment, so not you, but you heard about it at the conference, the antibody against E-beta.
Yeah.
So it could be related actually to what you said today. Okay, we cannot inject the LP1 antibody because it's risk for liver disease and all the other kind. But if we have these antibodies against E-beta, it's actually not transported anymore back from the periphery, probably. That's why it works. Yeah? Because it didn't.
Wait a second. Whoa. I don't understand. So you're saying the anti-A-beta antibody is not transported?
So, I mean, if you inject anti-A-beta antibodies in the blood, then A-beta only in the blood, but not in the brain is targeted. That's why it's not transported into the brain.
It is transported.
It is?
Yeah.
Oh.
I don't know.
Yeah,
They
the
find it in
question
the
was
brain.
if in liver there are other receptors that can
Yeah, I think the problem is might be the later stages when these animals develop plaques, right? Then, as I said in the very beginning, maybe the blood-brain barrier is altered. So then it's open, and then the antibodies can pass. I don't know how they would get into the brain when it's still tight, but apparently they get in.
Because the antibody itself doesn't go. How does it go together with
No, it does. I mean,
So not in this, yeah, it does, but not in these modes that,
apparently it's enough.
how does it work then?
I don't know. I mean, I was like you, when in 1999 the paper from Dale Schenk came out, from Elam, and they showed they inject an antibody into a mouse to treat Alzheimer's disease. I said, this is the most stupid experiment you can do, because antibodies do not cross the blood-brain barrier. And the plaques were gone, and the animals were normal. It works.
So then any antibody that's we get injected as a therapy goes into the brain as well, yeah?
Apparently.
Oh, that's fine.
Apparently.
Yeah.
And I mean, these studies have been repeated now with, I don't know, 20 different antibodies. They all work in mice. Right? And now some even in humans. It goes completely against the textbook. I have no clue how it works.
One more question?
Sure.
I should know this, of course, but I still don't. About the LP1, like B, as it called, expression button. Where is it expressed?
In endothelial cells or
No,
general?
just in general.
Where LIP is expressed?
Not LP, but LPB.
Oh.
LP1B.
1B. Well, pretty much the
You're
same
always
area.
short.
It is
But
expressed
it's
in
not really
endothelial.
taking over the function of LP.
No, it's not taken, but it is there too. But
But it's there too.
But it's only 30% of, for beta being transported by LIP1. So maybe the other one has 10 or 15%, I don't know. But it is there too. And all the ABC transporters have been shown to be involved in transport too. So they play a role as well.
Other question?
Yeah.
With a question, you spoke about injections of antibodies into the bloodstream, and what about injections into the spinal cord fluid, or was that from other any known results?
not yet. Yeah, we want to do that.
That's good.
But the problem is, we can only cure mice with that. We cannot cure humans with that, right? Would you be willing to that even modified antibodies are being injected into your spinal cord?
Well, again, it depends. It's very unpleasant procedure, but still, it's better than
Yeah,
that.
you have an immune reaction.
That's a problem.
Yeah, that's a huge problem. But mice? Well, you haven't done it, but we want to do that. I mean, we are just in the process of characterizing these antibodies, and the one I've shown you there was the one that we have characterized 2 months ago. Now we have one that's even better in blocking, but
Healthy mouse.
yeah, we get many healthy mice, yeah, which is good though. But no.
Okay.
Yeah, but if we know the epitope and when we characterize these antibodies better, we can even, you're right, I mean, we can think about then of modifying these antibodies and humanize these antibodies. But that I can't do, that somebody else has to do that then. But it would be possible, I agree.
I have maybe a little stupid question, but we know that the most common first symptom of the Alzheimer's disease is memory loss, because Alzheimer's disease affects hippocampus. So, why Alzheimer's disease affects hippocampus firstly?
It's actually not the hippocampus that is first.
But why?
It's a severe, you have a severe phenotype in the hippocampus, that's correct. But you have, it starts in the striatum, right? And then it goes in the hippocampus. But why? I don't know. It's a great question, it's not a stupid question. I have no answer to it.
Maybe hippocampus, maybe...
It's an evolutionary phenomenon.
But maybe the blood-brain barrier in the arteries of hippocampus have the lower resistance or something like that.
Yeah, I have to give you an answer to that, right? I mean, how can we measure that? I don't know. If there would be some specific protein that in endothelial cells in the hippocampus that we can tackle, then we could approach that problem. But I have no clue how to do that. And I don't want to. So maybe you got the impression that only clearance across the blood-brain barrier is the reason why you get Alzheimer's disease. That's certainly not the case.
Oh.
Okay.
I got it.
But I don't know how to specifically target areas in the brain in regard to the blood-brain barrier. I don't think that is possible, at least not yet possible. For us possible, we can't do that. I have no clue why it's the hippocampus so severely affected. Of course, you have expression levels. Think of the enzymes are pretty equal all over the brain. Maybe they're upregulated a little bit, but maybe the neurons are more, can be, the toxic effect of the beta is just more severe at the hippocampus. Maybe the neurons behave a little different. I have no clue. I have really no answer to that.
And maybe you said that the soluble A beta is toxic, but insoluble forms of A beta are less toxic, yes?
That's the overall idea that most of the people share.
Maybe hippocampus accumulates more soluble A beta than insoluble.
That is, in the ratio, certainly correct, because you have more plaques in the cortex, for example, right? So maybe you have more soluble there. Yeah, it's possible. It's possible. But again, hard to prove. I mean, then you could take out the hippocampus and mince it up and measure the amount of soluble beta. That is has probably been done, and I just have to check the literature. But I could check that. That's true. I could do that.
That's good.
Yeah.
So think of this antibody therapy. So you should inject it in quite a long period of time, yeah? So quite in advance in order to get the effect. So it wouldn't work if you inject already if you have a disease. So if you inject for quite a while, at some point you will develop anti-drug antibody. So the antibody against the antibody and it wouldn't work anymore.
Well, wait a sec. Which antibody are we talking about? The A-beta antibody? The A-beta antibody. I think they didn't inject it for a long time.
They didn't.
I don't think so. I think it was only a few administrations.
Yeah, that is good. But which time point? When it was already a disease?
You know. I don't know.
Yeah.
Check the website of Biogen. They have it in there, and they have a patent on it, so you can check even the patent. They will state the protocol there.
Okay. Any further questions?
Yeah, there's one more.
Oh, okay. It talks about an accumulation of A-beta around the blood vessel and around the blood-brain barrier, and that it can be transported from the brain through the blood-brain barrier, but actually is it transported in the neuronal cells or glial cells or within the neuronal tissue towards the blood-brain barrier? Soluble or unsoluble?
Yeah, is there a guided transport of the beta peptides
Yes.
through the blood-brain barrier?
Yes.
I don't think so. I think it's more or less diffusion. What's happening there? Yeah, I would say that. Because I don't know any, I mean it's soluble, right? It's out of the cell. I don't know whether there's any guided transport in some certain direction.
Yes, but this story about the accumulation of A-beta in some forms around the blood vessel, it's strange, you know. If there is no actually guided transporting or no reason behind the...
Yeah, but plaques are just accumulations somewhere in the brain, right?
Yes.
Because some peptides were sitting there, and then you have a seed, and then more.
Yes, but there must be actually the reason for this tendency to accumulate right around
That
the blood vessel.
Yeah, that tendency I can give you, because when you have a regular blood vessel, you have LRP there, right? An A-beta peptide comes along, it's caught by LRP, transported across the blood-brain barrier, it's gone. If LRP is not there anymore, comes along, sits at the blood vessel, stays there. Another one comes, finds another A-beta peptide, seed,
I know. Oh, okay, but it must get stuck to the blood vessel.
Somehow.
Somehow, yeah. But you still have to have the reason, but because if it's not got stuck, you know, there is no reason for it.
There is. I think nobody knows why a plaque is developed at that neuron and not at that neuron, right? And I mean, it's absolutely not clear why plaques are at specific places and not at other places. I agree. There has to be something, but it could be a certain amount of irons maybe, or that they're stuck there then maybe.
Yes.
But I don't think that has ever been measured.
It's a pity.
Can you once more name this interested antibiotic all cells except
Puromycin.
What?
Puromycin.
Puromycin.
We use that as a marker, for example, when you have a gene of interest and you want to express that in a cell, and you make a stable cell line, right? You have your gene of interest, then you have puromycin next to it, you transfect the cell, and then you add puromycin to the cells. And all the cells die, except the cells that have stably inserted that construct into the genome, because they have a puromycin and cassette
This is a stabilizer, yes, for the cell to not to be killed of puromycin.
No, the endothelial cells have PGP. They throw puromycin all the time out of the cell. So they can survive.
Oh, and a question a little apart from the topic. Maybe we can use pyromycin in oncology to kill this stuff?
Tumor cells? Yeah, but you kill the body as well. It's so unspecific. I mean, it's like, you in oncology you use Taxol, right? That pretty much kills all the cells. It kills the tumor, but kills everything else too. So that's the reason why you can use it only for a short time, and then the person has to recover.
Okay, this is non-specific at all.
No, non-specific at all. The only thing that survives with pyromycin is the endothelial cells. And,
More questions? No more questions? Well, we have enjoyed the course not less than you, and we're very thankful to you and to Dina also, who was the person who made an original invitation. And it feels like you've inspired a lot of thoughts and, activity that we'll see what it will lead to. Since we have a lot of students, maybe this will bring a good start to your projects. Thanks a lot.
Thank you.
Thank you.
So if anyone wants to work on Alzheimer's disease and has some questions even in the future, don't hesitate to contact me, write me an email or phone me, whatever. Ask